Hi, good morning, everyone. I'm going to talk about CO2 as a driver for neurofluid flow, and then specifically CSF. So, as Liliana already nicely explained to you this morning, CSF flow is very important for brain clearance, and this is important to maintain a healthy functioning brain. And she already explains the different pathways of CSF. And when CSF either enters or exits the brain, it passes by the located here next to the cerebellum. And in order to clear the brain, it's important that CSF moves throughout the brain. So how does it move? Jan already touched upon this a little bit, that there are CSF motion due to the heartbeat and due to respiration, which cause changes in cerebral blood volume, both on the arterial and as well as the venous side. And these changes in cerebral blood volume cause CSF to move due to these Kelly doctrine that states that a change in blood volume should be accommodated by an opposite change in CSF to maintain a stable intracranial volume. But apart from these variations in blood pressure changes, there's also vascular response, which causes CSF to move. So these are a couple of very interesting papers which looked into these driving forces of CSF. So, for example, neuroactive due to neurovascular coupling or vasomotion. And these papers, or one of these papers, also suggested that blood gases might be a cause for CSF motion, and then specifically carbon dioxide. So CO2 is a vasoactive agent, and it causes changes in the cerebral blood volume. So again, this will then induce cerebral CSF flow. And that actually brings me to the aim of our study, was to see if we could find a direct link between these changes in cerebral vascular response and CSF dynamics in the fourth ventricle by controlled manipulations of arterial blood gases. So how did we do this? We retrospectively analyzed all data that we acquired from an ongoing clinical study. And this all data was acquired during a gas challenge using a respiratory system. Here you actually see the paradigm for this gas challenge. Yeah, so this paradigm consists of a hypercapnic block, followed by a hypercapnic ramp, and then eventually a hyperoxic block. And all these events were interleaved by a 120-second baseline period. And the patients included in this clinical study, there were actually patients presented with brain metastasis undergoing radiotherapy, and they were all scanned here in the hospital. And this patient cohort was actually very interesting because they showed varying amounts of edema. And due to that, that led us to actually a second aim for the study, mainly to investigate whether changes in brain mechanical properties due to the formation of edema could affect the vascular response and also potentially influence the CSF flow. So we had data available from 22 patients, of which 12 of those were actually also scanned post-radiotherapy. And they were scanned on a 3T Tesla scanner prior and 3 months after radiotherapy. As I said, scanned here in the hospital. And the blood data was acquired with a TR of 1,050 milliseconds and a total of 5,000 volumes. And the planning of the blood data was aimed to cover the full brain. And that meant that in some cases, the fourth ventricle was actually included in the lower slices of the blood volume. And this was very convenient for us because this meant that we could actually image the CSF flow as well in those cases. So how does that work? Liliana already explained this a little bit, but if you use BOLT, you can also image CSF flow. So there's like a fresh inflow of CSF in the bottom slices. And these spins haven't seen all the RF pulses yet, so they're not yet saturated. So this will lead to a high signal intensity. And you can see an example here. Here it's actually CSF flowing in from the top. So in our case, it will be inflow from the bottom, but the principle is the same. So here again, you see the planning for this particular patient. And what you can see on the right is in blue, you see the hypercapnic challenge. And then following this hypercapnic challenge, you will see CSF inflow. So in purple, you see the signal as measured in CSF voxels in these lower slices. And what you can immediately already see is that the CSF inflow follows the hypercapnic periods. So then onto the vascular response, because we're also interested in that one. So how did we analyze that? Well, here again in blue, you see the hypercapnic block. And for this analysis, we only looked at the hypercapnic block instead of also the hypercapnic ramp, because we were really interested in immediate changes following these immediate drop down of CO2. So as I said, in blue, you see the hypercapnic block, and in red, you see the corresponding volt signal. And then what we did was we used this method by Poulin. And what it does is here again, you see the CO2 trace in black. You can ignore the dotted lines, those are not important. And what we do is we convolve this CO2 trace with an exponential decay, or actually multiple exponential decay functions with different time constants called tau. And then you end up with this convolved function. And what we did then was do a least squares fit between the volt signal and this convolved function. And then you end up with a certain tau fit, which you see here in yellow. And this tau fit is represented by tau, which actually represents the vascular response. Then for the CSF dynamics, in this case, we also looked at CSF inflow following the hypercapnic block, just as we did for the vascular response. So here in the background, you see again the volt signal, and in purple, you see the CSF inflow following the volt signal. What we did was fit a normal distribution function with a certain skewness to this CSF inflow signal. And then we took the half width at half maximum from this distribution. And as we're only really interested in what happens after the volt signal returns to baseline, we only looked at the half width at half maximum for the second part of this CSF inflow curve. So in total, we were able to see CSF inflow in 17 patients out of those 22. So in 5 of those patients, it wasn't, CSF inflow wasn't visible. This is probably due because it was a retrospective analysis, so the planning wasn't optimal. So we're actually very lucky to catch CSF inflow in those 17 patients. And here you see the results of those 17 patients. So what you can see in the top is again the hypercapnic stimulus and the hyperoxic stimulus in green. And here in red, you see the volt response. At the bottom in purple, you see the CSF flow measured in voxels containing CSF in the lower slices. What you can immediately see is that this negative change in CO2 is followed by an inflow of CSF. So there is a change in cerebral blood volume, which you can see from the derivative of the volt signal. and that will be followed by an inflow of CSF. And we also had this hyperoxic stimulus. And what it nicely shows is that although you get a bolt response from the hyperoxic stimulus, it doesn't give you a CSF inflow effect. So this actually also works as kind of a validation, because it shows that we are actually measuring CSF inflow instead of just venous bolt responses, because else we would have also seen a signal here due to the bolt response induced by the hyperoxic stimulus. Then you can also, as I already said, nicely appreciate the matching of the timings between the different signals. So in the dashed lines, you can see that the change in cerebral blood volume nicely matches with the time that the CSF happens. And for the vascular response, remember we had this very heterogeneous patient group showing very different amounts of edema loads. And one thing important to mention is that we focused on presumably healthy tissue, so we excluded all the edema regions. And what we saw over there is here you see results from 4 different patients. And on the T2 flares, you can see the amounts of edema, where the upper 2 patients have very little edema and the lower 2 patients show relatively a lot of edema. And it's also, you can also see that here depicted in these graphs, how much edema was found in each patient. And here in the middle, again, you see in blue the hypercapnic block, and in red, the bolt response. And in orange, you can see the tau fit that we did. And what we noticed in these patients was that an increase in edema causes this slower bolt response in these patients. So the tau was actually a lot higher, meaning that the return to baseline took a lot longer in these patients. And when looking at the CVR maps of these patients, we also saw that the CVR was decreased in patients with more edema. But note that we also saw this kind of mixture effect in CVR. So there were some regions that showed negative CVR as well. And when we compared the tau, which should represent the vascular response, to the amount of edema, what we actually saw is that there was this positive correlation between tau and edema. So what you see here is every dot represents a data set. So it could be either data from the post or the prior radiotherapy scan. And if there's a line between the 2 dots, it means that that data comes from the same patients and prior and post radiotherapy. And in orange, you see patients that actually had a recovery. So there was less edema found in the post scans. And in blue, you see patients that showed more edema found in the post scans. But what you can tell is that for most patients, when there was an increase in edema, this also meant an increase in tau, so a slower vascular response. And then for the analysis of the CSF dynamics, what we did, we looked at the CSF inflow following this hypercapnic block, so only the first CSF inflow peak. And what you can immediately tell is that if you look at those upper 2 patients again, the ones that showed very little edema, is that you get these nice sharp inflow peaks. Whereas if you look at the other 2 patients, the one that showed relatively more edema, is that you get this, also lower peaks. So if you look at the numbers on the y-axis, you can see in the bottom 2, those are actually lower, and we also get this widened or broader peak as well. Unfortunately, when we compared these inflow dynamics, so remember that half width at half maximum to the amount of edema, we couldn't find any significant correlations. but that might be due to the fact that we had this suboptimal planning and we only saw CSF inflow in 17 patients. Then onto the conclusions. So we did see a link between CO2 and CSF inflow, which is consistent with this Monroe-Kelly doctrine that, well, CO2 causes a change in cerebral blood volume, which is followed by a change in CSF flow. And what we also saw was changes in mechanical properties due to the formation of edema actually are correlated with a slower vascular response. So changes in the way the vessels react. And potentially these changes in brain mechanics could also lead to changes in CSF flow, but unfortunately, as I already said, we couldn't find that within this current patient cohort with the current methods that we use. So that was actually it for my talk. Thank you for listening.